Buongiorno a tutti, c'est tout for my English. My Italian, I'm very happy to be here, and I start this presentation with some slideshow in my way in science, basically from Middle Asia to Mediterranean. So,
Naila, sorry, open the Microsoft. Thank you.
in Middle Asia, in the nice lake of Istanbul, which is a really a good place. It's like Switzerland. Some people call it Asian Switzerland. And there was my first passion to be in the sea. But I studied in Moscow, in Moscow Aviation Institute as engineer in electronics and cybernetics. And this was good training for me, but was interrupted by, during this time I spent some time on the sea because I like it very much. But then my student life was interrupted, not student, my engineer life was interrupted by recruitment in Soviet Army, where I served in nuclear rockets, and then I decided quit this field and moved to physiology. So, and I worked several years in physiology by learning by doing. First of all, building electronic equipment to record activity of single channels in living cells, and this work was very efficient, and I was invited to Heidelberg to a laboratory of Bert Zachman, who afterwards won Nobel Prize, and it was a really productive time for me because we looked at basic properties of main neurotransmitter receptors like glutamate receptors, NMDA receptors, AMPA receptors, and kainate receptors. We looked at structure function, and it was hardcore basic research. After that, I moved to Amsterdam and became professor, and there my interest was shifted to more clinical aspects of brain functioning. So I will talk about it later. And finally, I moved to Marseille, which combined my interest in science and in sea, and there I met Francesco, and he invited me to give a talk here. And I'm very glad, as I said, and I now do some remarks. So as you can see from the map, there are many points which I visited and where I lived, and is there any pattern? And if you look at my educational background, it's like a pattern of a rocket, but my interest would be more patch pipette, which I used doing science, and in particular measuring current voltage relations in living cells like neurons. And this is combined my education and interest scientific. And now I switch to the main topic of my lecture, where I would present some examples based on our work, how knowledge of basic properties of neurons and their receptors may help to medicine. And I start with Fragile X syndrome. As you may know, that this is one of the diseases which is caused by inactivation of fragile mental retardation protein, FMRP, and this has severe consequences in neurological consequences and physiological consequences. So people suffering from this syndrome have mental impairment, attention deficit, and different other parameters of autism spectrum disorders. And they have also change in their appearance. And one of the property which is impaired in Fragile X syndrome was absence of long-term potentiation. Maybe you know that long-term potentiation is a type of synaptic plasticity which underlies, which is considered as a cellular substrate of memory. And in Fragile X mice, we used knockout mouse, Fragile X knockout mouse, which is a good model to study this disease in animals. And as you see here, the LTP in neocortex, in particular in prefrontal cortex, is impaired, and this may lead to some of the mental disabilities of the Fragile X patient. So to understand mechanism and in particular to find the way how we can ameliorate this parameter, we used slices, brain slices of Fragile X and wild-type animals to look at the alteration in synaptic plasticity and the ways to rescue this deficit. So what we did, we studied, as I said, prefrontal cortex, and in particular the phenomena called spike time independent plasticity. As you may know that to induce LTP, you need stimulate synapses heavily to be able to calcium ions enter the cells, which then make increase of the synaptic response. And the spike time independent, there are many mechanisms of LTP induction. The very famous tetanus stimulation, it's high frequency 100 Hertz stimulation, which probably does not exist in real time, in real life. So the spike time independent plasticity, which includes activation of presynaptic and postsynaptic at certain time interval, which may also increase calcium entry. As you see here, if you have just presynaptic excitatory postsynaptic potential and calcium, it gives some calcium entry. If you give backpropagating action potential in dendrite, it also opens voltage-gated calcium channels and gives calcium entry. But if you combine it in certain time delay, you may increase this calcium entry and after all increase in synaptic response. So we use this paradigm as more natural paradigm of LTP induction and compared this in wild type and Fragile X mice. And as you can see from these slides, in wild type, this protocol where you first stimulate presynaptically and then postsynaptically, you have LTP, which lasts for several minutes and even longer. But in Fragile X mice, this LTP with this paradigm is abolished, as was found for different types of LTP. And this is summary data. And what we found that the deficiency in LTP induction was due to deficiency in calcium entry using this protocol. So we played with different possibilities how we can increase calcium entry, and then if you make this protocol with postsynaptic stimulation, not single stimulation but a train, you can increase calcium entry, and then this protocol allowed us to have LTP in Fragile X mice, meaning that apparently more extensive stimulation of neurological pathway in fragile X may rescue of LTP, and this might be the way of treating this deficit. So after some analysis of literature, we found that one of the mechanisms which may provide excessive stimulation of sensory pathway would be enriched environment, and this what we used in our case. So we put mice in a cage with many toys, and after 2 or 3 months spending in this enriched environment, we looked at the LTP in these mice using the same protocol, and what we found that in mice which were in enriched environment, we could restore LTP. And this indicates for us that knowing this deficiency, neuronal deficiency in particular region of brain, and finding the way to rescue this deficiency might be good strategy for fragile X treatment, in particular enriched environment, which makes these mice more like normal. And this can be used, of course, for human therapy. And the next disorder which I would like to present, it's tuberous sclerosis complex. We heard just a few talks before about this disease. I just quickly remind that this is a multi-system disorder which affects different organs: the skin, kidney, lungs, heart, and also, of course, brain. And the most severe manifestation of this disease in brain is epilepsy, which appears in childhood, and 80% of these epilepsies are observed in children before age of 3 years old of age. So it's really early epilepsy, which indeed makes the life of children very, very suffering. And we have good connections with clinicians in Paris, and they always complain that this is epilepsy which they cannot treat properly because it's pharmacoresistant, and the only way to treat epilepsy is to remove tubers which appears in this disease, and these tubers thought to be the origin of epilepsy, and the main clinical treatment was to remove these tubers, and this may help patients. But in 30% of cases, even removal of these tubers does not help to treat epilepsy. And this is just to remind you that TSC, tuberous sclerosis complex, appears after inactivation of TSC1 or TSC2 genes, which are in mTOR system, and they participate in cell proliferation and growth, and when they are inactive, the mTOR system becomes overactivated, and this produces all these difficulties in health. And rapamycin is a substance which can block mTOR system. It works as a negative regulator of mTOR system. So this is just to remind you, because I will show you some data with rapamycin treatment. So, and keeping in mind all these difficulties, we started to study mechanism of epileptogenesis in TSC, starting with heterozygote mice in which TSC gene was inactivated. And these mice didn't show any morphological alterations in neocortex, no tubers, but we found that in 70% of mice, they showed spontaneous, non-provoked epilepsy when we recorded intracranially with extracellular electrode spanning all the cortex. You see here these epileptic discharges which were lasted for several hours. And this is an example of individual seizures at different layers of cortex. You can see they are quite long and quite severe. And even in hippocampus, you can see seizure events. And for our surprise, it was in the mouse, as I said, which does not have tubers. So meaning that tubers are not really not necessary to induce seizures. So to go into deeper analysis of these mice, we did coronal slices in TC animals and looked at the properties of neurons in different layers of neocortex. And in particular with interest in layer 4, because layer 4 is an acceptor of all sensory information coming from thalamus. And in these slices we cut so that all thalamic inputs were cut off. So we wanted to see whether this epileptogenesis is really cortical event, doesn't come from deeper layers of the brain. And indeed, in slices, when we use multi-cell patch clamp recordings, we saw aberrant behavior in neurons in different layers. You see here the bursts of electrical activity, which is reminiscent of epileptic seizure in slices. And if you compare the initiation time of the burst, you see that layer 4 is bursts in layer 4 precede bursting of different layers. This allowed us to conclude that layer 4 is initiation of region of epilepsy initiation in this model. So to understand what is specific for this layer, we looked at spontaneous postsynaptic currents mediated by NMDA receptor in particular, because recent studies, several studies show that there is change in expression of different subunits of NMDA receptors, AMPA receptors, but NMDA receptors are might be more relevant to epileptic phenotype, and because they are first numerous, there are many types of subunits of NMDA receptors, and they have slower kinetics of the current compared to fast excitatory AMPA current. And as you can see here, this kinetics of the current is different between subunits. And what is interesting that in previous studies, the people showed upregulation of NMDA currents, NMDA receptors with slower kinetics, 2G and 2C subunits. And so we looked at and these subunits have different kinetics and also magnesium sensitivity. And they are very good candidates to be pro-epileptic if they are expressed in excess. So we did recordings of postsynaptic spontaneous currents and concentrating on the late phase of this current, which reflects kinetics of NMDA receptor. And these results showed that in TSC mice there is slowing down of the kinetics of NMDA-mediated current, both in layer 2-3 and layer 4. and this is quite consistent. And this may, we checked whether indeed we have increased expression of these receptors doing real-time PCR, quantitative PCR, and indeed results show that there is indeed upregulation of GLU-N2C NMDA receptors, which has slower kinetics. And what is interesting that when we treated mice with rapamycin, which is one of the substances used to treat epilepsy in these patients, this treatment with rapamycin, it's 1 week intraperitoneal injection of rapamycin before experiments, showed that there was no increase of GLU-N2C expression, as shown here by red bars. So indeed, this upregulation of GLU-N2C receptors is mTOR dependent. So to look at electrophysiological consequences of blocking these upregulated receptors, we use different specific antagonists of GLU-N2C receptors, like UBP and DQP, and applied to the currents which we recorded in layer 4, for example, and as you can see here, that application of these drugs, both UBP and DQP, returned the kinetic of TSC mice to the wild-type kinetics, suggesting that this antagonist can rescue upregulation of contribution of upregulated GLU-N2C subunits. And this was again specific, mTOR specific, because mice treated with rapamycin, they didn't show this slow component. So both using PCR and electrophysiology, we showed that there is mTOR dependent upregulation of slow GLU-N2C mediated NMDA current. And this might be pro-epileptic because, as I mentioned, that layer 4 cells are cells which receive information from sensory information from thalamus, and then they are interconnected to each other and they serve as integrator of sensory activity. And if it's altered, it may produce a pro-epileptic effect. As you can see from this experiment, if you stimulate record from 2 cells simultaneously and stimulate 1 cell with a train of action potential, you see postsynaptic response, which fastly decaying in wild-type mice. But when you do the same in TSC mice, you see that there is still slow component which is left after end of stimulation. It means that in these mice you have a longer window for integration of sensory inputs. And this prolongation of this decay time was specific to, sensitive to GLU-N2C blockers. So basically this gave us like hint that if we block GLU-N2C receptors, we block integration, we reduce integration window which was enhanced in TSC, and then it might serve as anti-epileptic treatment. And indeed, in vivo studies, we used intraperitoneal injection of GLU-N2C specific blockers, and they reduced frequency first of epilepsy, and then even in some cases it blocks completely. As you see here, this is each square is an event of seizure, and then injection of UBP or DQP reduces seizures or eliminate them at all, suggesting that this treatment might be really anti-epileptic. If you do it in control animals or inject animals with vehicle, the seizures last longer. So this data showed us that this upregulation of gluon2C NMDA receptors in layer 4, in particular in layer 4, may have proepileptic effect in TSC, and blocking these receptors would rescue epileptic phenotype. And further studies show that this may be true in humans. So we did several experiments in tissue which we obtained after operation of TSC patients, and we analyzed this tissue with PCR, and again we saw upregulation of mRNA encoding gluon2C subunit. We've found that EPC, NMDA-mediated EPC is sensitive to gluon2C NMDA receptor subunit, and this, and also paroxysmal activity which we observed in several slices can be ameliorated by application of UBP. Here these are field recordings. You see spontaneous activity which in the presence of UBP becomes more rare, and this is another experiment where we see that the spontaneous bursts are reduced in the presence of gluon2C blockers. So this all shows us that understanding of the mechanism which may have proepileptic effect may help us to find the strategy and therapeutic drugs which can be used to treat this disease. And next disease which we studied was autism. We also use animal models. We used rats which mothers were injected with valproate, and the babies had autistic phenotype. And also we used Fragile X mice, knockout mice, which also have autistic phenotype. And this, to go deep into the mechanisms underlying autistic behavior, was first initiated by observation which clinicists made treating autistic people. They found that if you treat autistic children with diuretic bumetanide, you may ameliorate some scores, autistic scores, which have autistic children. So children were treated with 1 milligram per day of bumetanide, and you see if they had placebo, there was no change in the score. Here the lower score is the beta. So this probably you know what does it mean, like as clinicists. And see that bumetanide after treatment of 90 or 120 days reduced these scores, meaning that the autistic phenotype was ameliorated. And to understand the mechanism, I have to mention that the bumetanide is one of the blocker of chloride transporter in cells. So as you may know that chloride gradients are changing during development in cells, being high at the earlier ages, then reducing to lower level, and this chloride concentration is controlled by cotransporters, NKCC1 and NKCC2. NKCC1 is responsible for import of chloride, and it is active first. So at the beginning of development, it's even from embryonic state, you have increased chloride concentration, which makes GABA receptors, which are in adulthood inhibitory, to be excitatory. So at the beginning, they mediate excitatory synaptic transmission. But with development, KCC2 receptor start to be expressed, and this is exporter of chloride. So it decreases concentration of chloride when it's active, and this makes GABA receptors inhibitory, as they are known in adulthood. And this pattern is quite robust and found in many species. And one interesting observation for this developmental change in chloride was that during birth, if you look at these curves, during birth there is abrupt decrease of chloride concentration, or reversal potential of chloride through GABA receptors. And this was published many years ago, and this mechanism was assumed as protective mechanism during the birth. So during the birth, you have to inhibit all neuronal activity, and if GABA was excitatory, it would be a big stress to the brain, and nature developed so that during first hours before birth and few hours after birth, the chloride concentration abruptly decreased, and GABA receptors become inhibitory, so they silence all neuronal activity. And we looked at this phenomena in our autistic models of valparate mice and Fragile X, and what we found that this abrupt shift during the birth is abolished in autistic animals. And if you look at this curve, you see that the level of chloride remains almost constant, even when in control it goes down. So it means that these chloride transporters are not functioning properly, or one of the transporters, like KCC2, which is involved in export of chloride, is not functioning as it should be. As a result, you see that at P0, where immediately after birth, where in control it should be inhibitory, GABA is excitatory. And this you see by application of GABA receptor agonist. You see increased frequency of spontaneous activity in valparate mice and Fragile X, but not in wild type. Here it's decreased. And this indicates that chloride homeostasis is not functioning properly in mice which are models of autism. And this is example of other experiments where again we show that this aberrant activity is due to chloride homeostasis, because bumetanide improved all these parameters. And I would say that in this treatment we treated mothers before they giving birth with bumetanide. So 2 days before giving birth, mothers had bumetanide in water, and drinking this water helped to restore normal chloride homeostasis. And the effect was also acute, was present also when we applied bumetanide acutely to the slices. And another parameter which we looked at in autistic mice, there was increased spontaneous glutamatergic activity, indicating that a neuronal network in hippocampus in Fragile X and valproate treated animals is altered. So they have more excitatory drives, and which is not normal, and bumetanide again can restore this phenotype. And finally, some behavioral tests which we used, like vocalization. You know, when babies are taken from their mother, they still communicate by ultrasound frequencies, and you can estimate the parameters of their communication, and you can see that bumetanide restores all parameters like duration of calls, chevron or downward calls to normal. And also in vivo, you can see that increased activity in autistic models can be rescued by bumetanide. And this is good news, because these data show what might be the mechanism of autistic manifestation, and because bumetanide was known long ago like a diuretic, it is already in market as a drug, and all tests which are necessary for new drug development are already there, like toxicity, survival rate, and so on. So this drug can be readily used to treat autistic children. And now this bumetanide is on the third phase of clinical trials, and I think soon it will be maybe used in the market. And some people, some parents of children already start to use it as volunteers, and they have very positive comments on the behavior of the children. So this is like I showed you 3 examples how studying deep mechanisms, which you can do only in animal models, may help to find drugs for treatment. And to, just to summarize what I said, that basic science may give to medicine understanding of cellular and molecular mechanisms in neurological disorders, develop rescue strategies for treatment, and finding, selecting, and testing drugs for targeted pharmacology. And I think testing is very, how to say, is component which cannot be readily used for human patients. And with animal models, when they have properties of human disease, may be good tools for testing drugs and optimizing treatment. And to finish, I just want to show our team in MED. So we are now continuing studies of these disorders in different aspects, including role of inflammation and second hits on these diseases. And I hope we will have some more data which can be used in clinics. Thank you for your attention.
Thank you, Dr. Burnashev. The presentation is open for comments, discussion. I think you gave a very clear message at the end about the mechanisms, the basic science, providing useful information in 2 models of human. Do you have any? Please.
So first of all, thank you, Professor, to be here and to have accepted my and our invite. And just one thing, I just would like to say that it's really impressive how just differences in currencies in channels can change not only our neuronal activity, like, I don't know, seizures and epilepsy, but also, for example, create autism. And so really thank you for this. And I would like to ask you, you talked about UBP, so that can act as an antagonist for the GLU-N to CD channels. So if you have to answer to your French colleagues, the doctors, the pediatricians, that ask you about treating tuberous sclerosis complex, are you positive that in the future, in the next future, we can use a kind of drug like this just to treat these children that suffer really much? Thank you.
I think what I can tell now, we did this project together with clinicians from Paris. Maybe you know Olivier Dulac. He is very enthusiastic. And the compound we used is UBP and DQP, which is second one is more efficient, as you could see from data. And now we are collaborating with a group in the United States which made more selective and more efficient GLUN2C blocker, and Dulac is going to push it into clinical application. But it, as I said, with bumetanide it's faster, but for DQP it would take like 7 years minimum to pass all tests. And
Professor Kosarica.
Thank you for the presentation and the amount of data. You have shown that mTOR is in some way involved in the pathogenesis of the phenomena you showed. Do you have any experience on, for example, mice which are double knockout for ATG genes, those who are related to autophagy?
no, we are going to address this question, how mTOR, modification may affect expression of NMDA receptors. But we so far did not go into different models of that. But this is a good question. I think, but mTOR system is involved in many processes. I mean, if you look at other diseases, always mTOR is involved. Some of the elements of mTOR system is involved. And we are going to address this, but not in the way to answer your question now.
Now, I can't resist to ask Professor Nichelli, who is our neurologist, to give a comment. I mean, the entire lecture was focused on basic research impact in neurological disorders, and also the original presentation. Would you like to give a clinical perspective from the practicing?
Well, the basic idea that came from your very nice presentation is that basic science and clinical work are really close together, and you demonstrate very nicely that basic science can have an impact, a very strong impact on clinical work. And I would also say that the two parts of academia need to work together, because also the clinical work gives, I think, to basic science the idea and the hypothesis to work on.
Yes.
That's
Yeah, this
only
is what with bumetanide. It came from clinical application. They found that it, and then we went to see what is the mechanism.
Yeah, while you are getting the microphone, I would say exactly the same. We have been, in my area that is pulmonary, we have been disappointed, let's say, by animal studies. And we have eventually found that the best animal model is man.
Yes.
You know, and my question to you, do you, apart from the application of drugs that, like you have presented in your original and your, do you ever work with human tissue? Do you ever work with patients? I mean, do you take also tissue from patients
Yes,
and try to reproduce the
Yes. I
phenomena?
showed maybe missed for TSC.
Yeah.
We had several
Example
patients.
of
We took tissue from patients after operation, immediately after operation.
So
They
you
cut
do it also.
slice and also showed increased gluon2C expression, sensitivity to gluon2C antagonists, and reduction of paroxysmal activity in
Very
this
good.
tissue by antagonists of gluon2C.
very good. Next.
Hi, good afternoon. I would like to ask about the first part of your presentation about fragile X mental retardation. We have seen that LTP problems are related to poor stimulation, and if we put mice in rich environment, we can improve this parameter in the neuronal stimulation. So I would like to ask to you if there is any clinical possibility to apply this concept to real patients and to stimulate them with play therapy or other therapies by neuropsychologists to improve their outcome in the pathology?
I don't know exactly whether it's in use, but our data implies that this may help. If you may overstimulate some regions of the brain, like prefrontal cortex, you may improve outcome. But what we also show that if you put animals in a rich environment, you may improve it like without any pharmacological therapy. And I think it may be applicable for human, for sure, for these people who suffer from Fragile X syndrome. And I think it may apply also for other autistic disorders.
Thank you very much for joining us today. If there is no desperate need for another question, I think that some caffeine in our brain will be helpful at this stage. Thank you very much once again for your...
Thank you.